All right, so for this week, for your discussion board post that you'll do, just in a couple of sentences, kind of like reflect on your time in 214 this semester. And, I know we had this crazy transition, but, just tell us, did you learn anything? Did you feel like you were able to learn based on what you were given? Maybe what's your favorite thing that you learned? Just different things like this. And your GSAs will be reading through those. So yeah, and that will be the last discussion board that you will have to do. As far as homework, your homework for exercises 17, 18, and 19 will be due on Sunday night, same thing, to your GSA. So just make sure that you don't forget that. And then, yeah. Let me see, there was something else I feel like I was going to say. Oh, review session. So a lot of people in my other lectures yesterday said that they would, that a review session would be really great. And I had some people email me as well and say that would be really good before the practical. So there will be a review session. Okay, that's going to take place on Monday night at 6 p.m. Okay, it'll be on Zoom, so you use the same link, same meeting ID. Okay, however, there is a 100-person capacity for these Zoom meetings. All right. But I don't think that'll be an issue just because there's never been 100 people at my review sessions before during this semester. So of course that's optional. You don't have to come to that, but I'll just be going over some things for the practical. Okay. And then your practical will be posted on Tuesday. So that will be posted for you on Tuesday, and it'll stay up until the next Sunday. Okay, so you have from Tuesday the 14th, I think. So Tuesday the 14th to that next Sunday, whatever that is, to complete that practical. And then once you complete your practical, y'all are done with lab. Lab ends a few weeks earlier. They end other classes every semester. So you just take your practical and you're finished. All right. And so, your paper should, be graded within a week or so. Maybe give your GSAs a couple days of grace on that, just because they will be having to read all those papers and go through those. All right. But, then your practicals, those should be back to you relatively quickly, because there will just be, some questions that will be automatically graded by Blackboard just based on the type of question that they are. Okay. Yeah. So like I said, after that, you're done. Some people have asked me about extra credit opportunities, and I haven't decided yet. I need to just go through and look and see kind of how people are doing and kind of what things are looking like right now. And that'll kind of determine what what I'll plan to do. However, there could be an extra credit assignment, or I could have some extra credit questions on the practical, maybe both. I'm not really sure. But if I do offer that, you'll have plenty of time to do it, of course. And so that shouldn't be an issue for you. Okay. So any questions at this point about any of my announcements so far? If you have questions, you can just type them in the chat, and then I will get to those. Okay. So, like I said, your Quiz 8 assignment is posted on your Blackboard under course content, under quizzes. And I also posted Word documents of pictures of the anatomy models that you have to know for the practical. Okay. So there's two posts for that. One is the models labeled. So I have the models on a Word document, and I went and I labeled all of them for you. And then I have one that's unlabeled in case you want that to study from as well. Okay. So those are both up there on course content, and all of the pictures that I'll be using of the models will just come from that document. Okay. There won't be any new pictures or any new models. Okay. Everything will come, from that. Just make sure that you're reviewing the sheet brain dissection is not on there. Okay, so make sure you watch the video that I posted on YouTube, and check out the picture that's at the end of your PowerPoint on that we did a few weeks ago. You know, that's on Blackboard. But pictures of things from the cow eye dissection are that on that document. But I also have a video posted that I linked on Blackboard for that as well. But again, all of the recorded lectures, all of the videos of me going over models, and the dissection videos, those are all posted throughout your Blackboard announcements. They're linked to YouTube, so you can just check those out. I would definitely recommend doing that as you are studying. Okay, so if there are no questions at this point, you can still ask at any time. That's totally fine. I'm gonna go ahead and pull up our PowerPoint. We'll do our lecture. It shouldn't take me too long because it's not like a super long lecture or anything like that. Okay, so let's see. All right, so exercises 17, 18, and 19. All right, so let me, all right, so first we are gonna start with the eye. So we're gonna start with eye anatomy. We're gonna do external, then internal, then we'll do ear anatomy, do a little bit of physiology for that, and then we'll talk about taste and smell, okay? So that's kind of our plan for today. So this will be a good picture for you to know. There are a lot of pictures throughout this PowerPoint, and so I'll tell you if those are ones that you need to know or not, okay? So this is definitely one to know. All right, so we're gonna go through some of these terms, and I'm not gonna point out everything, but we're gonna go through some of it, okay? So it's not labeled really on here, but our superior and inferior eyelids are going to be known as palpebrae collectively, okay? So next we have our iris. And so our iris is gonna be the colored part of the eye that we see right here. And there's a set of two muscles that are gonna be found in the iris, the radial and the sphincter muscles. And the function of these muscles is to enlarge or dilate or constrict the pupil based on the amount of light that's in the environment. So the pupil is really just an empty space that allows for light to flow into the eye, okay? So if there's more light in the environment, then your pupils don't have to be as large because they don't need as much light in them to be able to see what's going on, okay? So in lighter environments, so if you walked outside like today here in Lynchburg, like it's really sunny, so your pupils would be smaller, so they'd be a little bit more constricted, okay? But if you were to walk outside at night, or if you were to be in a dark room, then your pupils would be much more dilated because they'd need to let a lot more light in from the environment so that you would be able to see, all right? And so another structure I want to mention that's not actually labeled on this picture, it's called the cornea. So it is labeled on your models, okay? And there are two functions of the cornea. This is not on your PowerPoint, so definitely make a note of these. The two functions are to bend light and to protect the interior of the eye. Okay, so the cornea is going to lay over the iris and the pupil to protect that part of the eye. Okay. All right, so next we're going to talk about tears. All right, so let's start with kind of where they begin and kind of where they end up. Okay, we also have this flow chart here on the side. That's going to be really important. You want to make sure that you look over that. You will see that again. Okay, so first we have our lacrimal gland right here. So that's where our tears are going to be made. These secretions will be made in the lacrimal gland. And then we see these little lines coming off of it. That's to represent the lacrimal ducts or excretory ducts. And so when our tears are made in the lacrimal gland, they're going to flow through the lacrimal ducts and they're going to flow over the eye or down our face, depending on how emotional we may be. Okay, then they're going to go into they're going to drain into the lacrimal caruncle, and then they're going to be going through the lacrimal canal. Okay, once they hit the lacrimal canal, they'll be drained into the lacrimal sac. They'll flow down the nasolacrimal duct and end up in their nasal cavity. So when someone may be crying a lot of times their nose will get kind of runny. And so the reason is because all of those tears are flowing down through their nasal cavity. All right. And so it's those secretions that are coming through the nose. Okay. All right. So in the eye, or on the eye, excuse me, we have 6 different muscles that you want to know. I also have a video going over the eye model, talking about where the different muscles are, how to determine left and right, medial and lateral, things like that. So definitely make sure to check it out. Okay. So we have our rectus muscles and our oblique muscle. So our superior, inferior, medial, and lateral rectus, and our superior and inferior oblique muscles. So our superior oblique muscle will always be located medially on the eye. So we can see that right here, and then right here. And then our inferior oblique is located laterally on the eye. Okay, and so looking for the superior and inferior oblique muscles will help us to determine which side the medial and lateral rectus muscle is on. All right, because like I said, we can tell the difference between medial and lateral sides of the eye. All right, so you don't have to know this picture, but all of these structures, well, most of these structures, you will be responsible for knowing on the eye model, okay? So we have 2 cavities in the eye. We have an anterior and a posterior, and the anterior is going to be filled with aqueous humor, and the posterior is filled with vitreous humor. And so you can kind of see those represented here. But for example, on the cow eye, when I do the dissection and you watch that video, you can see all of the posterior, you can see the vitreous humor kind of coming out of there. There's a lot. The whole cavity is kind of filled with it. And it's this gelatinous or jelly-like kind of material. So yeah, so that's kind of what that looks like. And then we have these different layers of the eye. We call them tunics, but they're kind of like tissue layers, okay? So the most superficial is the sclera. So that's the fibrous layer. So when we look at our eye and we see the whites in our eyes, that is our sclera. All right, just deep to that, it's this little red layer right here. That's the choroid. And so that's the vascular layer. So we have lots of blood vessels there. And then lastly, our deepest layer is the retina. So it's in yellow right here. And that is our neural layer. All right. So talking about a few more structures, right here we have our lens. And so our lens is what kind of allows our eyes to focus so that we can see clearly. All right, and our lens is held in place by these ciliary bodies and these suspensory ligaments. All right, and so they kind of hold it and they allow it to change shape to focus. All right, so right back here is kind of where our optic nerve begins. So it's that neural layer kind of converging to form the optic nerve, and we also have some vasculature right here. But at this spot where that converges, that's going to be our optic disc, and so that is our blind spot. We call it our blind spot because we have no cones or rods there, okay? Our cones and rods are going to be our photoreceptor cells. So they're going to be what is allowing us to see. Our cones allow us to see color, and our rods allow us to see in dark lighting, okay? We also have another structure related to those photoreceptor cells right here, the fovea centralis. And right there is where we have the largest concentration or density of photoreceptor cells in the eye. Okay, so we have a lot of cones and rods there. All right, you don't have to know this picture because everything I'm going to ask you about external anatomy of the ear, I can ask you on the model, okay? But I'm going to tell you what to know, and I also go over this in the video as well. Okay, so for external anatomy of the ear, you want to know the helix, So it's kind of this top part of the ear right here. The fossa is just kind of this area, also known as the scapha. You can say either one, that's fine. And then the lobule or the earlobe, okay? We kind of remember from earlier classes that most of the external ear is going to be made up of cartilage, except for our lobule, that's going to be made up of adipose tissue. All right. And then right here is going to be our external acoustic meatus, external auditory meatus, or external ear canal. Any of those will work. But I, again, wouldn't ask you to label that right here or on the model, okay? If I was gonna ask you for that, I would have this part right here labeled, okay? All right, so we're kind of moving into the middle ear, and I'm gonna talk about kind of how sound will travel through the ear and kind of what's going on so that you can kind of hear and interpret sound and kind of, you know, what that means, okay? So our ears are uniquely designed to where the shape of them is going to help guide sound waves into our ear canal, okay? So sound waves hit the ear and they're guided into the ear canal, okay? And so then these sound waves are going to come down the ear canal and vibrate the tympanic membrane, which is our eardrum, but we want to say tympanic membrane, okay? Hits the tympanic membrane, and then it's going to go through our ossicles. So our ossicles are these tiny little bones in our ears. All right, we have the malleus, the incus, and the stapes. The layman's terms for them are hammer, anvil, and stirrup, but we want to know the anatomical terms, okay? So the sound waves will then kind of pass through these ossicles, and then they will go to the cochlea, okay, which is this kind of snail-looking structure, and that's in the inner ear. So once these sound waves or vibrations reach the cochlea, we have this like kind of subpart of the cochlea called the organ of Corti, and it's like this, the curly part of it, so the little circular part right here. And I kind of outlined it in red for you right here. So the organ of Corti is what is actually detecting the sound, because it's going to kind of pick up on the stimulus of those sound waves, convert that to action potentials, and then send it to the brain by the vestibulocochlear nerve. All right. We also have a couple of other structures that help us with equilibrium and balance, and those are the semicircular canals and the vestibule. All right, the vestibule helps with static equilibrium, and the semicircular canals help with dynamic equilibrium. And we also have these structures called otoliths in the vestibule that kind of help out, kind of as like a supporting role, okay? So next we're moving on to taste. Okay, so we kind of know what the tongue does. We know that it helps us to form words when we're talking and having conversations, and it also helps us to move food around while we're chewing and swallowing. Okay, we have 5 basic tastes. We have sweet, salty, sour, bitter, and umami. Okay, and umami is like a savory type of taste. Okay. I put this little graphic in here for you because this is, and you would do this if you were actually in lab. There is an exercise where you would like take different tastes and use a Q-tip and like dab your tongue with it to see like where you could pick up those tastes. All right, and so this is like the taste bud mapping, where supposedly these taste buds are for these different types of taste, okay? However, a lot of research has been done to show that this is not accurate, and that there is not a specific location on the tongue where the taste buds for different tastes are, and that they're just scattered throughout. However, this is in your lab manual, so this is what I have to teach you, okay? So just know this, you'll probably see it come up on your test. All right, and then next we have these different types of papillae: the filiform, fungiform, circumvallate, and foliate, okay? And so these are shown on this picture right here on the right, shown where those are located. All right, and those are kind of associated with some of these different areas on the tongue. But really these papillae function just to help with, you know, when we're chewing food, just to kind of move that food around pretty much. So it's just not like sliding all over the place in our mouth. All right, so we have this little kind of step-by-step like pathway, I guess you could say, of taste and how we interpret that. So we have these little taste pores on our tongue, all right, and they have these little tiny little hairs called gustatory hairs that are extended into them, okay? And so when we're eating, when we're chewing, all right, chemicals in the food will bind to those gustatory hairs, and that will trigger depolarization, so an action potential of the gustatory receptor cells. And that stimulus that information is going to be transmitted to the brain by cranial nerves 7, 9, and 10. So the facial nerve, glossopharyngeal nerve, and the vagus nerve. Okay. You won't have to know this picture, but you do want to know this pathway right here. And lastly, just a little bit about smell. Okay, so all I really want you to know is that we have we have a lot of different olfactory receptors, and they are really unique for a specific reason. Okay, this is not a PowerPoint, so write this down, listen up, whatever. Okay, so our olfactory receptors are unique because they don't just bind one type of molecule. They bind a whole class of molecules. So molecules that are kind of similar in structure or different properties that they have. Okay, so it's not just like a one-to-one ratio relationship, okay? They can bind a lot of different types of smells, okay? And so that's why we can smell new things and smell lots of new things. And tell when something is new that we have not smelled before, okay? And guys, that is it for our PowerPoint for our lecture today. So I'm going to go ahead and stop sharing. And then if y'all have any questions about announcements that were made about the material, you can just type them in the chat box. I'll give y'all just a minute for that. All right. But as always, don't forget, please email me or your GSA if you have any issues, if you have any questions, or whatever the case may be. All right. It does not look like there are any questions. All right. So I am going to. Oh wait, here are questions. Okay, so for the practical, it'll just be what we've covered since the last practical. So exercises 12 through 19. 12 through 19, yep. And yeah, sure, Grace. So the olfactory receptors are unique because they can bind to whole classes of molecules. So you don't just have like one receptor for one smell. You have one receptor that can pick up on a lot of different types of similar smells. Oh, Kayla, thank you so much. That's so sweet. Yeah, it's been so crazy. There's been so much going on, and it's been a lot for me to get used to. And I know y'all weren't in my class, at the beginning of the semester, but I've had fun with everyone and, getting to know y'all. So, yeah. All right. Okay, it doesn't look like there are any more questions, so I'm going to go ahead and stop my recording for this lecture today.